Thank you. So despite what this first slide says, I do actually have a conflict of interest that I have to disclose, which is with GSK, who I have a small amount of research funding from, but it doesn't affect the content of this talk. So I'm going to talk about some of the methods for solving the crossing fibre problem that Donald talked about. Here's a quick overview. I'm going to start with a bit of a recap of the limitations of DTI, which Donald covered. Basically, I'm going to use that discussion to motivate some of the objects that we try to estimate using the methods that I'll talk about in the body of the talk, which comes next. So I'll talk about the kind of standard methods out there for studying and resolving crossing fibres. It's not a comprehensive review by any means. I'm going to focus just on the basic methods and give some pointers to more sophisticated refinements of those techniques. And I'll finish with some discussion of the key applications of those techniques and some open research questions. So this slide highlights the limitations of the diffusion tensor model. So there are various kind of simple configurations of fibres in that this column on the left. Now remember that these axon fibres are much smaller than typical voxels in MRI. There might be hundreds of thousands of them in a single voxel. So they can take all sorts of weird and wonderful configurations. And these things are really actually quite simple configurations, but they are the kinds of thing that we do see in the brain. So the second column here then shows the distribution of particle displacements or scatter pattern that we expect to see within each of these configurations. So this is the fundamental object that diffusion MRI measurements are sensitive to. And you can see that the configuration of fibres affects this distribution of particle displacements within. So for example, these straight parallel fibres cause a scatter pattern which is elongated along the direction of the fibres. These fanning and bending configurations cause a more widespread pattern because there's distributions of orientations which push particles in different directions. And then these crossing configurations cause more complex scattering that have these ridges along the directions of the different populations of fibres. So as Donald discussed, diffusion tensor imaging models this scatter pattern with a Gaussian distribution. The Gaussian distribution in 3D has ellipsoidal contours. So the basic assumption is that this scatter pattern is ellipsoidal in shape. And here in this third column I'm showing the best fit ellipsoidal shape to these scatter patterns in the second column. So we can see that in these kind of simpler configurations with one sort of central direction, unimodal configurations, the ellipsoidal diffusion tensor model is a reasonable description of the scattering that we expect in these configurations. But for these more complex scatter patterns that we get at fibre crossings, the ellipsoidal model is just not flexible enough to capture that added complexity, and it discards a lot of the useful information that it contains. So let's think about what it is that we'd really like to be able to estimate from diffusion MRI measurements for doing things like tractography and connectivity mapping. What we'd really like to know is how many fibres in each voxel are pointing in each individual direction. And that's what this idealized object in the fourth column represents. So this is something I'm calling the fibre orientation distribution function. It's a function of the sphere, because each point on the sphere represents a unique direction, and the value of the function at each point, or for each direction, is just the fraction of fibres that point in that direction. So for example, for the straight parallel fibres here, most of the fibres are pointing in about the same direction. So we get a large peak in the fibre orientation distribution function in the central direction of those fibres. Now we get some slight width to the peak because these fibres wiggle about, so the orientation changes slightly along their length. Then for these other configurations, the fanning and bendings, there's a planar distribution of orientation, so we get these paddle-shaped peaks, and the crossing configurations have peaks in the directions of each of these distinct orientations. So then the last two columns are showing the signal that we measure from in diffusion MRI, from which we want to estimate this kind of object. So in this column here, I'm showing the signal as a function of direction. So I'm assuming that we make this kind of a familiar spherical acquisition scheme where we measure a single measurement in lots of different directions but with the same B value. So we can see that in these kind of unimodal configurations, generally the signal is low where the fibre orientation distribution function is high. But for these more complex crossing configurations, we can see that the things aren't quite as simple as that, because in fact for this orthogonal crossing we get maxima in the signal along the directions of the peaks of the fibre ODF. This other object here is just minus the log of the signal, so this is the apparent diffusion coefficient in each direction. Again, it looks like the relationship is simple between this function and the fibre ODF for these unimodal configurations. The peaks are in the same direction, but looking at these crossing configurations, we can see that things are more complex, because now the peaks in this orthogonal crossing are in the intermediate directions between the peaks of the fibre ODF. So the point is that we have to work quite hard to get from the things that we measure, the signal here, to the thing that we're after, this fibre ODF. So here's the sort of broad classes of method that I'm going to talk about for doing that. So the first class of technique I'm calling multi-tensor model, so Donald covered this to some extent, and this is slightly distinct from the other ones in that it just measures a discrete number of fibre orientations, whereas the other three classes that I'll talk about all try and recover a function of the sphere that in some sense reflects that fibre orientation distribution. But let's start with these multi-tensor models. So the broad idea is that there's some number of distinct populations of fibres within a voxel which have their own orientation, and we can model the scatter pattern in each of those populations with a tensor model. So then the sum of the Gaussian distributions that those tensors represent give us an overall scatter pattern, which is the kind of thing that we expect to see within this kind of configuration. Now, as Donald said, we have to work a bit harder to fit these models to the data, and these can cause various kind of instability problems, but there are ways around that by constraining the solution. And we can get some sensible output from these things. So here's just an early example showing a crossing fibre region where we basically get a mess from the single diffusion tensor model, but using a two-tensor model in the region of the fibre crossing, we consistently resolve the orientations of the crossing fibres, which allows us to track through that crossing. But this slide does highlight another problem with these techniques, which is that we kind of need to know how many fibre populations there are in each voxel in order to be able to pick the right model to fit. So there's a model selection problem. And there are various ways in the literature for solving that problem, but all of those introduce problems and limitations of their own. Related ideas, compartment models. So the simplest one of these is probably Behrens' ball and stick model. So this is the model that underlies the probabilistic tracking in the FSL package. Very simple model. So it assumes two different populations of water within white matter, some not next to the fibres or outside the fibres where diffusion is assumed isotropic, and another population inside the fibres where diffusion is considered perfectly anisotropic, and then the overall signal is the sum of those two. I mention it here because it extends easily to multiple fibres. We can just include multiple stick compartments. And then the charmed model is a similar idea, but they use more sophisticated models for the intra and extra cellular water diffusion.